So this is just a quick recap of the primer that I did a few months ago. We had this fun data set of cats and dogs, and we used a perceptron to classify them. And for the next hour, this is what a neuron is. It is just a weighted sum of input values attached to a nonlinear function. And if you want all the gory details of grinding through this algorithm to learn this decision boundary, you can watch the primer on YouTube. And it comes with this lovely animated GIF of learning the famous MNIST data set. And you see the numbers start to appear, but this is a shallow learning. So we're really just learning templates for each of those handwritten digits. And so each of the templates has to compete with the others. So where two numbers overlap, you see this kind of ambiguity. And these learned templates never really coalesce on the real number. Like this 9 is fighting the 8, which also has a loop at the top. And that motivates deep learning, because we cannot work directly in the input space. We need to learn features from the input in some embedded space, and then in that space draw our decision boundaries. And that's what we've been doing with our variants when we filter them with convolutional neural nets. So now we begin. So a little history of variant filtering. It's always handcrafted. It's just, what are you handcrafting? At first, we handcrafted the hard filters. So everyone picked their favorite number at which to cut off the good variants from the bad variants. And you could twist these knobs all you liked, but pretty quickly people realized there were drawbacks to this approach. Any number you were cutting off on was totally independent of other features that you might know about your variant. So these lines that we're drawing in our decision space are all orthogonal. They're just perpendicular to the axis.
Just to make sure everyone's on the same page, can you define variant filtering? What is variant filtering?
Okay, so variant filtering is the task, given a variant, say, is it good or is it bad? We'll look at some examples in great detail. True biological variation versus some kind of artifactual relic of the process that the DNA went through in order to be sequenced. Yeah, or it could be an artifact from library preparation or from mapping. There are lots of places where artifacts could have entered the picture. We want to separate those from the real biological signal. So the first way this was done was with these hard filters on summary statistics about each variant. So if, for example, all the evidence for the variant came from the positive strand, that sounds a little suspicious. So people would think of, okay, if 90% or more of the evidence comes from the positive strand, we're going to filter that out. And everyone had their favorite little set of features, but there were all these drawbacks that you weren't actually modeling anything. You were just cutting up the decision space, and you weren't looking at the annotations together. The next sort of step forward was to learn the decision boundary rather than just tuning knobs to find it. And there were a lot of algorithms in this space. The variant quality score recalibrator in the GATK, for example, uses Gaussian mixture models to model what real variation looks like in a high-dimensional space of these summary statistics or annotations. But you could also use a support vector machine. or random forests. So random forests are sort of like taking those hand-tuned hard filters and automating that. So you just generate a bunch of random hard filters and then learn which collection of hard filters work the best. But in all of these algorithms, we're working on handcrafted features. So we're learning the decision boundary. We're learning how much to weight each feature, but the features themselves are the secret sauce. They're what we think is a good variant, expresses what a good variant is, or what we think expresses something suspicious. The next step in handcrafting is to learn the features, machine learn the features, machine learn the decision boundary, and now we're handcrafting the architecture. So the crucial difference is we're giving much more to the algorithm to learn. Rather than deciding what the algorithm should get to see from the input data, we give the algorithm all of the input and say, you figure it out. What's the signal? And so in that way, it's sort of more open, more to learn, rather than just figuring out where to draw the line. And this, of course, foreshadows a day of machine-learned architectures over machine-learned features for machine-learned decision boundaries, at which point we can all retire.
So for the second one, presumably you also need more ground truth data, or it's more sensitive to that?
Not necessarily.
Okay.
Why do you think you'd need more?
Yeah. If you're throwing everything in, what are you throwing at it now?
Over here?
No. In this hypothetical, or maybe.
Oh, here you're saying you need more data.
Yeah.
Yeah, definitely. You need big training sets for deep learning. Yeah, absolutely.
Sorry, I guess that's the third bullet.
Yeah. Okay, now we're on the same page. Yes, absolutely. This requires much more data, because if there's some there's no one doing the sanity check on this architecture. So we wouldn't give a handcrafted feature that we knew was meaningless. But it's very easy for a neural net to pick up something that's a total artifact of the process if the training set isn't carefully developed and large. Yeah, absolutely. So it's a powerful model, but because of that, it needs much more data to be trained, so it doesn't abuse that power. Okay, so while we're taking a stroll down memory lane at the data sciences platform, or the DSDE, we used to have something called variant review lunch, where we wouldn't even have an algorithm do it. We, as humans, would sit and eat pizza and classify variants. So because this was so much fun, we're going to get to do a quick session right now. And while we do that, okay, so here's IGV. Here is a variant. What do people say? Good or bad? So now we're classifying.
Not everyone has seen this before, so can you explain?
Yeah. Okay. So here, how do we sequence the DNA? We get some sample, put some really high-pitched noise around it, shatter the DNA into these little reads, and then we put it in a Illumina box that reads the reads, and then we align it to the human reference. So over here is the reference sequence. Each of these little pointed worms is a read, something that came off the sequencer. Okay? Everywhere they have one of the bases. So this is going to be a little loud. But they all, right, have DNA in them. And almost everywhere, it's identical to the reference, which you see at the bottom. That's the human reference. Okay? But I'm going to turn off all the bases because that's a little loud. And we see these remain. Why do these remain here? Because those are not what the reference has at that location. The reference at that location has a T. But here we have a C. Okay? So we read a variant. And now it's our job as variant filters to say yes or no. Do we let this one through, or do we hold it up? I'm getting thumbs up, thumbs up, thumbs up. It looks good. Okay, why does it look good? It's a SNP, okay. About half, right? So we're a diploid organism. We expect to see either half of the reads supporting the variant or all of the reads supporting the variant.
Negative strand, well represented.
Beautiful. It's well balanced between the red and the blue. Nothing suspicious there. It's not coming from the ends of the reads. I mean, sometimes it is, but sometimes it's in the middle, so it doesn't seem like any kind of statistical bias there. I agree. This is a good, solid SNP. And
So now let's just do that a million more times.
and we're done, right? So, okay, this is, because we're going to be looking at these later, I want to get you used to it. Now, this is the same exact site, chromosome 7, position 1119, blah, blah, blah. This is the same site, okay? But displayed as a numeric tensor. So over here, we have it in IGV. You see that blue line of C's? Here is that blue line. You see how it's only half of them have this blue. It's just a different way of representing the same thing.
What is everything else? Are you going to tell us?
We'll talk about it, but I mean, most of it is the information that's available in IGV. So IGV here is showing you all the reference sequence, all of the read sequences, and there's metadata too, like how confident the sequencer was about each read, and the strand information. So all of that is encoded in this numeric tensor as just a sort of matrix or a high-dimensional matrix object that a neural net can process. So this is what the humans look at. This is what the neural net looks at. But it's the same thing. Okay? Let's go to the next site.
And the confidence comes out as the grayness of the...
Indeed, exactly. Yeah. So, view. Okay. Another site. What do people say? Good or bad?
What are we looking at here?
So now we're looking at chromosome 18, base 41 million. We've got a lot of reads. In the reference, we see GCA, GG, A, a lot of A's.
This one looks different.
This is not a SNP. It does look different.
A lot of people have never seen these pictures.
So, any thoughts? Well, first, what are we looking at? We're not looking at a SNP. What are we looking at? Okay. It's a deletion. Because these A's are missing. These two A's are missing from the reads. So this is how IGV shows deletions. And it just shifts the reads over so that they stay aligned with the reference sequence. This is a deletion. Is this a good deletion or a bad deletion?
It's pretty consistent.
Yeah.
It's like a homozygous deletion.
Yeah, exactly. This is a homozygous deletion. There's this one read that's a little suspicious, but the preponderance of evidence is in support, and the evidence doesn't seem too suspicious for any other reason besides we got one bad read. Okay? So I'd say this one can go through as well. And just for consistency, that same site as a numeric tensor is here. Okay? Whoop. And this is how the numeric tensor represents the deletion with this purple stripe, right? And see that's all those A's. That's why this is all green, because we have a string of A's, a poly A. But the reads had the deletion. Okay, next site. We got 2 more.
Could you, if you're going to show those pictures, could you give a little bit of a feel for what the indices are? I mean, just like ramble off a couple for this tensor. You have some multidimensional. Or are you going to tell us that?
We'll go into that.
Oh,
yeah.
great. Okay. Yeah.
Okay, next site. Here we are. Oops. Any thoughts? Sort by base. Nope. IGV can be a little finicky sometimes. Okay. Yes or no? Good or bad? Getting thumbs down.
Which one?
Okay, well that's part of the problem, right? But we're looking at this particular site, but people don't like it. What don't we like about it?
There are, clustered events nearby. But it's not just the way that it's not just that there are clustered events, but there are many different haplotypes represented, many more than 2.
Totally. This is not a diploid site. Something's going on here, but it's certainly not a simple SNP. And you can look at the ratio. It's definitely not 50-50 there. This is something we would want to block. The same site, this is that same site as a numeric tensor. And you see here it's much more clear. These are not 2 even haplotypes. These are totally different proportions.
What is going on there? I mean, like in biological explanation?
It could be a misalignment. It could be a copy number variation. It could be problem with the reference. Like there actually are sites that have this sequence, but they're not included in the reference. Regardless, it's clearly not a nice small insertion or deletion. Yeah, or SNP.
Can you blot each read? Blot each read?
We could blat these reads. We're not going to do that right now. Okay, and last site. What do you say, good or bad? At first, this one looks like maybe it's okay, but all of these reads that include it, that support it, you see, they just end in this poly A. They don't span across this poly A. So all these reads, excluding these that get it, all these aren't actually evidence because they just have A's here. And during our graph construction, we put in a deletion, but they're actually not supporting it. So there's very little evidence for this variant. Okay, so that's a little taste of a variant review lunch. We would all eat pizza, and we would go through these sites, and we had this baroquely complex database where you could enter the true status for different variants, and it was all tagged with the name of the person, and you could argue with someone. You could say, maybe M. DePristo thinks this is a variant, but M. Shan says, no, that's not a variant. And all this information was kept over time. And it's this really interesting data set, which has now been put to pasture.
So Sam, it sounds like you're using some expert knowledge about error modes here when you say there's a whole bunch of A's and I don't trust it because there's a whole bunch of A's. I don't know.
Yeah.
Is that?
I mean, that's what I'm using when I look at it.
Expert knowledge.
Well, some, amateur, amateur knowledge, professional amateur. I think we know that the homopolymers are very much associated with errors. So the context is definitely influential on how high quality your variants are.
Okay. So you're hoping machines can learn things like that?
Absolutely. But we need to give them the context for them to. But before we can even do that, we have to give them some training data. And with all those years of developing this human-curated database of good and bad variants, we had about maybe 2,000 or so, maybe 10,000 variants in the database, which is not enough to train a big, deep model. So we needed truth sets. And luckily, the government and Illumina also saw this need. And the Genomes in a Bottle project and Platinum Genomes were two big efforts to get a truth set over a whole genome of germline DNA. And these truth sets come with a confident region. So there are areas that are still very hard to call. We just ignore those. But for much of the genome, we have these validated calls. Okay, given a set of validated calls, which is my validated VCF here with these blue stars, I still need negative examples to train my model on. Where do those come from? Those come from an unvalidated VCF, that is just a raw VCF that has the good and the bad. And then I look, if I have a call in the confident region where there is not a corresponding call, that becomes a negative example. So now I have positive and negative examples with which to train my model. And there's just recently been produced this SynDip data set here at the Broad of a haploid organism which was sequenced with a long-read PacBio sequencer. So it doesn't have any of the Illumina artifacts. So another truth resource, which...
Not a haploid organism.
I'm sorry, hydatiform mole. So it's a pregnancy that the egg has no DNA and the sperm just duplicates itself. So you only have the... Is it? I'm pretty sure it's the sperm.
So what happens is that the sperm. So this was sorry to cut you off. Yeah, so this is something I worked on with Heng Li and Yossi and others. So the idea was to take these hydatiform moles. So this is an egg that in the mother, instead of the sperm's DNA entering and complementing the DNA in the egg's DNA duplicates. And so it becomes diploid, but fully homozygous and starts to grow. And this is not a viable pregnancy, but the nice thing is if you sequence this, it's going to be homozygous, which removes a lot of the error modes. You get very accurate sequence, especially on other technologies like PacBio, so you can get a full build from scratch, de novo build that way. And if you do this for two different lines, which is the novel aspect, you get sort of
that's the
synthetic.
So it's a synthetic diploid. But we know, because we have them as haploids, we know what, we're much more confident about what the two haploids should be. So this is a really great truth set.
So then you can see what real heterozygous calls look like in the data.
Right. So we mix the DNA and put it through the whole sequencing Illumina pipeline to get this unvalidated VCF where we knew exactly the sort of phase truth.
Exactly. Okay, so just going back onto how does the neural net see the DNA? Well, the simplest encoding is just this one-hot encoding of a sequence of DNA. So we have a channel for AC, G, and T, and every time we see that particular base, we put a 1 there, zeros everywhere else. Naturally, we can encode some kind of doubt. So if there's an N, then we can just like split the doubt amongst all 4 channels, or we can say, well, I know it wasn't an A or C, but it might have been a G or a T. We can express that ambiguity as well. But there's a lot more information besides just the reference sequence. We have all these reads. So Alex was asking about the axes in the read tensor. Those are 4 for the one-hot encoding of the reads themselves. But here we're not doing a one-hot encoding. We're actually doing a base quality encoding. So we encode the sequencer's confidence of that base at that site. And then we have the reference information. So that's another 4 channels. And then we have deletion or insertion, as well as a number of read flags. So which strand was the read on? Was it first in pair, second in pair? And the mapping quality as well. Okay, so quick quiz. Now that we're so good at looking at variants, what kind of variant are we looking at here? It's circled. It's a SNP, yeah. Heterozygous, homozygous? Heterozygous, great. The reference is these sequences. So you see how they align here? It's just a slightly different blue for the C and a slightly different green for the A. But they're stacked, so it's in 3D. So there's 8 dimensions of just sequence information between the reads and the reference. But it's all in there. What do we got here? Homozygous, heterozygous? Heterozygous deletion. And lastly, this one's a little hard to see, but we've got an insertion, a homozygous insertion. Okay, so these are our read tensors. This is that 3D volume. You see the reference here you're just asking about. It's all the same. So that's why it's vertically identical, because everywhere aligns to the same reference. But we expand it into this very sort of uncompressed representation so that we can do convolutions over this, so that we see that whole spatial structure, and we're going to convolve. So what does convolution do? Convolution in general is smoothing. If you do any random filter, it's going to smooth. But if you're very careful about the kernel you choose, then convolution has other properties. So see, a convolutional kernel is moving across, and now suddenly we get new behavior. Now it's doing edge detection. The edges are popping out of the image. Here we have horizontal edges because this is a horizontally aligned convolutional kernel. Now it's vertically aligned, and the vertical lines pop out. So this is the edge detecting property of convolution. For convolutional kernels, and now we're detecting diagonal edges. And now we're doing a Mexican hat. So it's like a sombrero, or it's the Laplacian of the Gaussian. So convolution has these great properties of pulling out edges, of selecting particular frequencies, and from a neural net's perspective, it allows us to decouple the size of our model from the size of the input, and it allows us to share weights across space. And this sort of encodes our belief that the input is translation invariant or translation equivariant. That is, if I see a horse's head over here, it's still a horse's head if it shows up over here. So the features I'm looking for in one part of my input are equally informative if they show up somewhere else. And convolution naturally expresses that. Okay, that's convolution over an image. What is convolution over a sequence? Well, it's a very similar thing. It acts as a motif detector. So instead of pulling out edges, we are pulling out motifs. So here, if we were to convolve this TC, TC, TC detector with this reference sequence, what will we get? So here I'm doing a one-hot encoding, but I'm just showing it with the color. So blue is C, red is T, G is brown. And that little black line is a very rough sketch of what the output of this convolution convolving this with this would be. So right here, you see in the sequence we have red, blue, red, blue, red, blue. That's TC, TC, TC. It found what it's looking for. That's a motif detector looking for TC, TC, TC. It finds it, it gets a big response. Just like the edge detector, when it saw a vertical edge, it gave a big response. Here, when we see a TC, 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 we get a big response. What types of position weight matrices do these convolutional neural nets learn? They learn all kinds of things that we used in our variant review lunch implicitly. They learn homopolymer detectors, so long stretches of one base. They learn STR, short tandem repeat detectors. So when we have a TC, 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 or a CCA, CCA, all that is very commonly associated with artifacts and is very useful to be aware of when you're trying to classify variants. They also just learn sort of these general properties of GC content in the model. Okay, so we've now got this ability to convolve over sequence. We have truth data, and now it's time to make some models. So the sort of simplest neural net to attack this problem, yeah? Yes. Well, 4 of the dimensions in the tensor are the reference. Make sense? Yep.
So you said that these convolutional neural networks are motivated when you have some kind of translational invariance. But you were showing in your numerical tensor that the potential SNP is always in the middle, right? So is that like problematic? Because you don't really have the translational invariance anymore?
Yeah, so that's a good point. We don't expect the SNP to, I mean, we sort of always feed the SNP in the center when we're doing variant filtering. The translational invariance is for motifs that might be informative about whether the SNP is real or not. So it's not going to help us detect to localize the SNP right where it is, but we're not trying to do that in classification. Later on, we'll look at an architecture where that is very important, but in that case, we don't center the tensor at the SNP. Okay, so this multi-layer perceptron, yeah, question?
Yeah, sorry. In the previous slide, you were saying, CNNs learn these kind of different types of detectors. Is that the CNN, when trained on the variant calling task, implicitly is learning these detectors?
Exactly. And we'll see that happen live if you just wait a few more slides. So here is the sort of equivalent to the previous variant filtering algorithms in that this only needs the variant annotations, the summary statistics. There's no structure here. There's nothing to convolve over. We just learn a multi-layer perceptron, like we did with the cats and dogs, except that was a single layer. Now we have multi-layer perceptron on the annotations. This is just purely predicting from the reference sequence. So it's one-dimensional CNN, but this is very handicapped, right? It's all it can see is the reference sequence. It knows nothing about the reads, nothing about a site of variation. But already this can predict far better than chance. So just in the reference sequence, you can build a model that is very informative, if not competitive with real algorithms. But there's tremendous amount of information in the reference sequence. Then you can combine this reference convolution with a annotation multi-layer perceptron we have this flattening operation where we throw away the structure, and then we just build a multi-layer perceptron on top of that. And this model is now available in the GATK. And it's very fast because this, again, is not looking at the reads. We're just looking at the annotation values associated with the variant as well as the reference sequence. And then these two architectures, which are more powerful but much bigger and much slower to compute with, use the read tensor. So they're convolving over that big 3D block that we've been looking at the GIFs of. Okay, so when I compare this with the DeepVariant paper, which was recently came out of Google Genomics, so they're not exactly solving the same task, but they are very comparable, and we'll show how they stack up. But first, just qualitatively, what's going on. So first, DeepVariant is a genotyper. This architecture is called Inception. It was developed to classify images, the ImageNet database of about 1.2 million images into 1,000 classes. It's a very innovative architecture because it has what they call these inception nodes. Each one of these is an inception node. And it does convolutions in parallel and then one-by-one convolutions, which are essentially learned dimension reduction. That's what's happening at each of these concatenations. So they were able to get a way more depth than had previously been possible to train without these inception nodes and the parallel processing. They also had these auxiliary classifiers to help feed the gradient back down deeper into the net, because the gradient, which is what we're going to use to learn these models, travels all the way from the last layer to the first layer, and it can disappear by the time you get over here. But this model was developed to classify images, and there were certain appeals to the statistics of natural images that occur in the architecture, namely this max pooling, which is where you just take the maximum response in a given area and throw away everything else. This makes it so you lose the precise neighborhood relation that you had earlier on. And you see right after the first convolution, there's a max pooling. For DNA, we don't have this idea of smoothness. The neighboring bases, I mean, sure, we have homopolymers, but in general, neighboring bases, you cannot expect to be the same as whoever is before or after them. Whereas in pixels, that's a really reasonable assumption. So max pooling is something we try to avoid in our networks or just use towards the end as we get deeper in after we've developed features based on this local coherence. Okay. This model, when it was first designed, had 1,000 neurons at the end, which were those labels of images. The DeepVariant paper rips out that last layer and replaces it with a 3-neuron layer that classifies heterozygous, homozygous, or reference, the diploid genotypes. All the work of, is this an insertion or a deletion, or where do the reads align, that's all handled by other algorithms that are not deep learning. So this takes aligned reads after local realignment and then genotypes them.
So you have biallelic polymorphisms, and that's it, and then you just follow each person.
Exactly.
So you're essentially saying that you translate the DNA region into an image, and then
yes. Right, and that's another big difference.
Network.
This is a 3-channel image. This is a RGB image. So instead of in the read tensor, which we have where we've expanded the channels, each base has its own channel. In the DeepVariant approach, there's one channel which encodes sequence information, another channel encodes base quality information, and a third channel encodes whether it matches the reference or doesn't match the reference. So there's big differences in the encoding, and there's sort of more technical differences in the architecture and in what we're actually trying to do. So we are filtering variants. In DeepVariant, they are genotyping. Regardless, each algorithm gives a score to a variant, and with a score, we can make comparisons. And that's what we show here. So we're comparing to VQSR and VQSR run on Nomad, as well as DeepVariant and the two-dimensional CNN. And this is area under the precision recall curve. And this is on a small test set of variants that were not shown to our model during training and that are balanced between positive and negative class. Yes?
Can you say a little bit about how you deal with differences in coverage? So when you're wiring up your network, do you just set to like the maximum coverage to be the size of your initial?
Exactly. So we have a maximum coverage here, and then when we have more than that, we just randomly downsample until it fits on there.
Is there any sense in which this is an unfair comparison in that the type of data you use to test is sort of similar in some ways to the type of data that you use to train yours, but not that the other models were used?
This is from running DeepVariant with their binaries on, like, it's on the same BAM, but that's what they expect to take as input. So in that way, like, they're making their own image from that BAM, and that happens in their code. That I don't know how it. I didn't write that.
Right, but the BAM and your BAM.
Same BAM.
Right.
So your model and DeepVariant are trained on data from the same data source?
So DeepVariant, I'm not 100% sure on their training methodology, but they trained on the NA12878 from the Genomes in a Bottle project on that BAM. But the model is available for use with any BAM. I mean, that's the idea. Yeah, so this model, so in particular here, we just trained on NA12878. And this model just trained on NA24385, the Ashkenazi child, and this was the CHM, the SynDip. And so these are three different true sets.
So you're telling DeepVariant and the GATK4 CNN were trained on the same data? That's it. Thank you.
Not always. Like DeepVariant was only trained on. So here, that's a fair assumption. But in the CHM, like DeepVariant was not trained on CHM.
So if you expect that if it was trained on the same data that the CNN was trained, it would do better than it did?
I think it would certainly do better if they expanded their training set. And that's something we see with our models. If we have a bigger training set, more samples, more chemistries in particular, we perform better. And I'm sure just training on 2 samples is not going to be enough. Yeah.
Did you try a sort of ensemble approach between DeepVariant and your CNN to see if...
I did not, yeah, but that probably would squeeze out some more performance. Even just doing a different random initialization, training the model, and then making an ensemble in that way, you usually get a better performance. Some more results. I'm so far behind time that I'm not going to even really talk about this. We've also looked at true set generalization. So train a model on the CHM, apply it on genomes in a bottle, etc. There clearly is generalization across true sets, but these true sets are vastly different in their qualities and in their quirks. So we're collaborating with Illumina to first build bigger training sets, but also to standardize how these evaluations are done. Okay, there's often complaints that deep learning is a black box or that it's alchemy and that we're just sort of throwing things at the wall and seeing what sticks. I sort of like considering it the land of the unknown prior better. That is, it's these tricks we have of dropout and batch normalization. They impose a prior on our data. It's just not entirely clear what that prior is. Another metaphor that I prefer is that it's like cooking. You must bring together these excellent ingredients. You follow a recipe, that's your architecture. You carefully wash your hands and prep your data in that you separate your testing, your training, your validating. You don't let them leak. You don't look at the testing data until you've already trained your model. You watch this gap between the validation loss and the training error loss, and you call it overfitting or memorizing. But with cooking, there's this magical moment when you put the thing in the oven, and something else takes over. And that's stochastic gradient descent, and we're watching that here on these ROC curves. We're tuning the parameters, and that's cooking it. So you see the model learn. But what does it learn? So this is a little bit more accurate representation of the motif detectors that come out from this variant classification task. This is sort of a bug. I didn't clear the screen between each batch, but I kind of like the Slurpee look. This is with updating it. But there are many of these kernels, right? We have maybe 500 in a model. And the model is learning the ensemble of this. It's not learning one particular motif detector. It's learning all of them and how they together help it classify variants. So here you can pull out, this is like a poly G detector. Got a poly A up there. What else is interesting in here? Got a little STR detector here. You see the blue and red. And there's some CG detectors. This is like a CG detector. Yeah.
All the types of detectors you just described are purely sequence, but you have other dimensions in your tensor which are like quality and strand and so on. So are those also kind of motifs being
Yeah, so
learned?
here we're just looking at the 1D CNN, which only had reference sequence. So these are just motifs learned on purely sequence. Yep.
And is there like a whole family of different detectors for deletions of different size, or how does that work?
Yeah,
Because
so
the invariance is gone.
yeah, so these kernels are just on the reference sequence. So this model is going to have no idea about deletions, but the 2D model will certainly pick up detectors, and I think we have some in here. This is like a CTT edge detector.
Even GC content, I have a hard time. I mean, there must be some whole bag of detectors that you can kind of associate roughly with picking up CG content.
Sure.
Is that?
Yeah, and I think so. These are just screenshots, but this is visualizations of the detectors learned in the 2D model, and you do see this. For example, this is like a deletion or the poly T detector or poly A. And these are sort of similar idea, but with images showing as you move to more abstract layers, you develop more and more complex features. More visualizations of image processing neural nets, sort of exciting the last layer, which is what we did over here. We really tried to excite a neuron and see what input would really excite it. So we get this sort of poly A thing or this poly T. For images, they're much more easy to interpret. You can sort of excite the frog neuron, and you get this frog-like thing, or exciting the cheeseburger neuron and getting this cheeseburger-like thing, though it's not so appetizing. Nor is it a cheeseburger. It's clearly not a picture of a cheeseburger. So these images are very important reminders that what the neural net has learned is not at all what we think of as the true label. So these are images, you show them to a neural net, they are 100% sure that this is a frog. But you show that to a human and we're 100% sure that's what a neural net thinks is a frog.
So
2 slides ago, those were partial derivatives of the output layer in terms of the input, or?
So you take an input, random initialization with the Gaussian, and then instead of taking the derivative with respect to the loss function, we take the derivative with respect to the activation of that particular neuron and update, and go through the same stochastic gradient descent process, but now we're just trying to optimize the activation of this. So it's a little bit sinister. You're trying to light up that one neuron. Yes. Yes. Okay, but sometimes these things aren't totally interpretable, and I've gone to great lengths to try to make these motif detectors interpretable to humans. So I've done something called spatial dropout, where we throw out whole convolutions, the output, which encourages the model to learn motifs that are informative, independent of what the rest of the network is doing. So this motif can't rely on some other motif, and they can't collude. So it's sort of Jeff Hinton via John Bloom motivates this as sexual reproduction, where you sort of mix all the DNA together, and that makes it much harder for pathogens to sort of memorize it, and it gives us robustness. So you can't have these long-range dependencies if you can't count on something in chromosome 12 always being there. Okay, but ultimately it's a distributed representation. It's not necessarily interpretable, and I don't think that's necessarily a problem. Like there's great confidence to be gained when we see something we recognize, but if it does the job, maybe that's good enough in certain cases. I just think we shouldn't. The ultimate barometer is whatever you set the loss function to. So if you don't understand what the model learned, that's your problem.
So that's because the task here is essentially measurement.
Yeah.
And the details of the nitty-gritty details of the error modes of these Illumina machines and the pre-processing of the strands into libraries or whatever is maybe we don't care about that.
Yeah.
But that's specific to this task. I think.
Yes.
And different from a lot of bio tasks.
Absolutely. And I just think we should be clear about that. Like sometimes what we actually want to learn is some interpretable fact. And then in that case, you want to set up your loss function so that that will come to the surface. But there are going to be times when these models learn things that are not readable to the naked eye, and that's not necessarily problematic.
If it is interpretable, it is a benefit if you have a model that isn't.
Yeah, and it gives us so much more confidence in what's happening when we can interpret it. So I'm being a little bit provocative in saying that it's your problem if you don't understand what the model learned, but I think there's something to that perspective as well. Yeah.
I have a side question. I don't know if it's time for side question.
Did you want to take it to the session?
All right. Okay, so I'm just going to skip that. And I'm going to skip this too, except to say we've really tried to optimize the hyperparameters, and this is stepping into that last layer of machine learning the architectures. So we generate tons of random architectures and see which one's better, or we try to do Bayesian optimization over these hyperparameters, take smart steps in architecture space, but it's this huge space, so it's very hard to take intelligent steps, even with Bayesian optimization. And one exciting avenue is this idea of residual nets. There's a beautiful implementation from the imaging platform of residual nets, which are a way of getting even deeper. So inception let us get deep, but residual nets let us get even deeper, like thousands of layers deep. And that is an exciting avenue for attacking this problem of vanishing and exploding gradients. We were going to hear from Jürgen Schmidhuber, who argues that intelligence is all about compression, but I don't think that's the whole story. Intelligence also generates, and when we classify something, we've compressed it. We took this rich representation of an image or a genetic sequence, and we reduce it to a single label. And that's sometimes brutal, right? We've thrown away all this beautiful stuff just to say there's a cat in the image. Well, show me the cat, you know? The cat's what I care about, not the label. And I think that we can make the same argument for our genomic data, is we really want to learn a good representation for it, but we don't want to just label it. We don't want to throw everything away. To that end, and there's some motivation for this in neuroscience, but I'm just going to skip that. To that end, we're now learning to segment the genome. So not classify. Don't just answer the question what. Answer the question where. Where does the event happen? Build a segmented map of the genome. And that's where I'll leave you. So just to clarify, this is taking in the same exact input as before, the read tensor. But instead of outputting just one of those 4 neurons, this is a true SNP, this is a false SNP, we now output a sort of strip as long as the read tensor was. So if we covered 128 base pairs in the read tensor, we output now 128 base pairs with the labeled genotypes, with our prediction for the genotypes. So this localizes the information. It doesn't just classify it. And with that, I'll leave you, or open up for questions.